Well, yours is mainly your coach, it's not really.
Can we talk about it? Can we talk about
I would have to get it. I feel like I get along well with everybody.
Can we talk about why she's so happy? I feel like we never did talk about it.
Seriously, she's so fun.
And it was nice. I was here. We never did it. I mean, we never even did it on the bench.
Are you sure friends or?
Yeah.
Oh, okay. Oh, I was gonna say.
That was something else.
You guys were.
Wait,
So, we no longer have to do, we no longer have to take the pictures.
oh, that we don't have to.
What?
Where's she going?
So we have to do both parts?
Okay.
She makes it. Oh, that's what
And I stepped together.
She sees Michael and, yeah.
I'm sorry. Did we actually do that handshake thing yesterday? Or did I do that? Or did we do that?
No, we didn't.
Say that I never had.
Look at how close they just also,
Yeah.
and why do you keep doing, why do you keep changing the dance class so that,
Yeah.
like, yeah, you
know my dance. I'm gonna be so honest.
me either, she can't watch video, but you can't because no one knows what they're doing.
I'm like on something. I feel like doing that. I just stay out all the time.
And that's it.
There.
I feel like you just push it and just like.
I do. And I'm just like, that's my thing every day. I look at people and I'm like,
360 and 360 down.
I forgot.
It is a 90 degree.
What?
Yeah, I'm thinking
Which is 90 degree, you get carrots.
What's a 90 degree?
I have carrots.
In heyday.
Oh.
I'm gonna go buy some.
I completely forgot about
We're
it.
doing that, we have to, for sure. Which,
I'll go to skate.
I have to do all these things.
Yes. Because it's a 2-hour drive and lanes are like, probably around like 6 or 7.
All right, guys. Happy Friday. Good morning. I'll quickly start by taking attendance, and then we'll go through the worksheet. We'll go through the answers, and then whatever time we have left, we'll continue with the last couple sections of Chapter 3. Riley. Brianna, A. Drew. Bria.
Here.
Henry.
Here.
Nick. Abby. Sorry. Hannah. Here. Rosa. Matt. Jillian. Mia. Annie. Brianna Hill. Brandon. Brandon. Sadie.
Here.
Jessica. Jesse. William. Marissa. Sophia? Matthias? Colin? Braden?
Here.
Amber?
Here.
Katie? Katie? Oh. Ben?
Here.
Kennedy? Kennedy? Ava?
Yes.
Kendall? Emily? Lauren?
Here.
Thank you. Jen? Jen?
Here.
And Isabel?
Here.
All right. Cool. So does everybody have the worksheet with them? All right. We're gonna go through the questions. I just, I'll have you guys answer them. I just don't want the same people answering all of the questions. So I do want to see, everybody participating when we do these things. All right, so number one, why do you think so many environmental microbes cannot be cultured in laboratory broth or agar media, or just in a laboratory setting? Yeah. Yeah. So for those who didn't hear that, we don't have the specific environmental and cultural conditions figured out yet to grow these types of microbes. So we know that they exist from sequencing efforts, but it's difficult to get the conditions perfect for the growth in a laboratory setting. Does that make sense? What are the differences between prokaryotic and eukaryotic microbes? Yeah, that's.
Prokaryotics are usually smaller, containing no nucleus, 16 RNA, and the majority don't in underground organelles.
And what about eukaryotes?
Eukaryote are larger, containing a nucleus, 18 RNA, and membrane organelles.
Okay. Everybody hear that? Okay. And then what are some phenotypic features that distinguish bacteria from archaea? Yeah.
I said that bacteria has like different amino acids and sugars in their cell walls, and they're chemically distant from bacterial membranes than archaea, and then that archaea is more similar to eukaryotes than bacteria.
Yep. Great. So although bacteria and archaea are both prokaryotes, archaea actually resemble eukaryotes a little bit more in terms of their gene expression machinery, their cell walls, their cell membranes. You don't have to be concerned with the exact details of how they're different just yet, but that's what you need to know. Does that make sense? What are the two requirements that need to be met in order for algae to grow? Brianna?
Water and sunlight, but temperature makes it grow faster.
Yep, water or moisture and sunlight, just like the polar bears. All right, what makes fungi stronger than steel? Yeah, the chitin. And what is chitin? Yeah, Ben?
It's a carbohydrate structure.
Yep, it's an amino polysaccharide, so it's basically a carbohydrate with an amino chain as well. And yeah, so chitin is what makes fungi really, really strong in their cell wall. Okay, questions so far? Okay, describe benefits to the plant and to the fungus in a plant mycorrhizal symbiosis. Yeah.
Fungus provides minerals and better water absorption for plant roots, and plant roots give it a steady supply of carbohydrates.
Yep. So for those who might have not heard that, fungi provides plants with increased mineral nutrients by extending the volume of soil that's accessible to the plant through hyphal transport. And the fungi also provides or improves the plant's access to water. The plant provides the fungi with carbohydrates due to photosynthesis. Okay, now switching gears a little to microscopy, what are the distinctions between detection and resolution?
detection is the ability to identify an object through a microscope, and resolution is the closest distance that objects can be while still detected as something.
Yes, exactly. Detection is the ability to determine what the presence or absence of something, and resolution is the smallest distance by which two objects can be separated and still be able to be distinguished. Okay. Summarize the use of microscopy among scales of microbial size. So just in terms of, what the resolution is, you don't have to memorize these, but just kind of understand an idea of what the human eye can see, and then light microscopy, the scale that you can see things at. What's the resolution of the human eye?
Like 0.1.
Yeah, 0.1 micrometers. And then light microscopy, what's the resolution limit? 200 nanometers. So those are the two that you should know. Okay. Describe the 4 different ways that light interacts with an object. Yeah. Yep. And can someone tell me what those mean?
Refraction is when light bends when it enters a substance and changes its speed. Reflection is the wavelength of light bouncing off a surface. Scattering is there's a small percentage of incident light that's scattered in all directions, and absorption is when an object falls apart.
Yep. Exactly. Did everybody get that? And which one is microscopy dependent on? Refraction. Good. Brandon? Make sure I get you down. Okay. Number 10, explain the relative advantages and disadvantages of a wet mount and a staged preparation for observing microbes. So what are the advantages and disadvantages of a wet mount?
You can see them in their natural state, and you can do that quickly, but a disadvantage is that some move too quickly to see, or the sample can dry out, and it's hard to tell between the cell and the background.
Perfect. And then what about a stained preparation? Yeah.
Enhances the structure of the microbiome, and they can also be stored for a long time. Limitations are it requires fixation during it. That's a little bit longer temperature.
Yep. Does anyone need me to repeat that? Describe the use of fixation in staining in bright field microscopy. Why do we fix things and why do we stain them? Yeah.
They're fixed to the slide so that fixation adheres the stuff to the slide so it's in a fixed position, and staining is so that we can better see the microbes.
Mm-hmm. Great. All right, what functions do membrane proteins serve? Now we're moving on to a new chapter. Yeah.
They provide structural support, detect environmental signals, secrete virulence factors and communication signals, and they're part of ion transport and energy storage.
Yep. So this would be a good, which of the following is not true about a function of membrane proteins type question. Yeah, so make sure you know those. All right. Any questions so far? All right, so explain how fluidity relates to kinks and cyclic structures within the fatty acids and phospholipids.
Kinks in the chain increase fluidity, while cyclic structures decrease fluidity, and unsaturated chains are more fluid.
Yeah, exactly. All right, draw the structures of gram-positive and gram-negative cell envelopes and compare and contrast them. We have a big whiteboard right here. Who wants to come up and draw gram-positive, and then gram-negative. You don't have to be an artist, you just have to have the key things in there. All right.
This is fun.
There you have some markers.
Oh, Leonardo da Vinci, it's gonna look great.
Let me grab you a marker. All right, there's just one.
You just imagine her writing. Just stand her awkwardly till she gets back. I could stand over here. I'm the blinded. Wow, that's really bright. I'm blind. I'm a professor now. Alright, cool. Good day so far? Eh? What'd you guys do last weekend? What are you gonna do this weekend? Anything fun? Any plans? I'm genuinely blind for this. This is terrible. Any fun plans? Yeah.
What are you doing right now?
Who's going to work this weekend? Raise your hand if you're going to work this weekend. 2, 3 people, 4 people. I'm just standing here awkwardly.
And put this on the opposite. Settle up.
There we go.
I don't know, but I kind of like it.
Boom. She gave me blue and red so I can make purple. Will not be doing that though. Alright, cool. Time to check out these art skills. Cool.
Oh, he's doing both, okay.
You're at negative and positive. Alright. We're just gonna pretend that continues going that way, because why not?
Okay.
Both of these are gonna have this structure. Does anyone know what this structure is called? It's the phospholipid bilayer. Ain't that cool? With our gram negatives, they don't turn purple, they turn pink, and you can go back to that side if you want. They're gonna have like 1, maybe 2 layers.
Ben, didn't you just ask for the picture?
I'm drawing the picture.
Okay.
Yeah, 1, maybe 2 layers of this peptidoglycan layer. And if they have 1 or 2, just to compare and contrast. And then our positives, there's like lots of layers of peptidoglycan. It's supposed to be purple, but I don't have purple, so.
Can you label it, right, peptidoglycan layer or PG?
Ooh, PG works for me.
Great.
Is that good enough?
Great.
Awesome, there we go.
Okay, awesome. So, thank you, Ben. That was very informative. Did Ben miss in his drawings? Yeah, do you want to go up and add an S layer? And like, what makes gram-negative different in terms of the membranes? How many membranes do they have? Yeah. Do you mind adding to that?
Here's the S layer. And the gram negative has
Yeah.
My drawing is shit.
Yep. Yeah. That's what I was looking for. Good job. Yes. Yep. Thank you. All right. So, I really like, you guys being involved and you guys teaching each other. So I'm going to try to utilize that more. But one of the key things is that the gram-negative cells have an inner cell membrane, just like the gram-positives, but they also have another cell membrane up here, and that connects through, this is called, what are these connections that connect it to the top? Yeah, lipopolysaccharides, LPS. So that would be this. Alright, does that make sense to everyone? And then a small amount of PG. There's gram-positives. Do gram-positives have an S layer? Yeah. Alright. Anybody have any questions on that? Why is peptidoglycan a good target for antibiotic therapies? New hand.
Peelle207-PC has enzymes that make for excellent targets for new antibiotic treatments.
Yep. That's definitely true. In the context of why do we target peptidoglycan in particular, can we add a little bit more to that? Yeah.
It's only found in the bacterial cell wall.
Yep. So peptidoglycan is only found in the cell walls of bacteria. We as humans, when we have an infection, we don't have a cell wall. We're not, bacteria or plants. So targeting peptidoglycan specifically won't hurt us, but it will hurt the bacteria. So it's, it makes for an excellent target, and, there's enzymes in peptidoglycan synthesis that are also important. We do not have a cell wall. So number 16, the 2 mechanisms of resistance, I did not go through that. Did you guys answer that or try to?
I looked it up, but
Okay, yeah, sorry about that. I forgot to remove that question. I was just, I talked about that last semester and I realized we were short for time, so I skipped over it. So we can ignore that one. What's the purpose of the S layer in prokaryotes? Yeah.
It is an additional layer of protection, produces cell shape, makes the cells not stress.
Yeah, so it's a protective layer common to bacteria. It's a crystalline layer of thick proteins that contributes to cell shape and helps protect the cell from drying out. All right, any questions on any of that? Okay. So generally, I won't collect this from you. It's for you guys to study from. If you're here and you're participating, I will, give you the points for it. But if you were not here today, then you should email it to me before class so that I can give you the points. Just an FYI. All right. So we'll move on and we'll go through some more on bacteria. This stuff is a little bit of a review. You should have already gone through bacterial cell division. I just want to kind of give you a quick review on these things, and then we'll talk about some specialized structures of bacteria. So overview, we're going to talk about the nucleoid, how bacteria divide by binary fission, and then some specialized structures. So we talked a little bit about the structure and function of the bacterial cell, starting from the cell envelope, and now we're kind of going to work our way towards the middle, and specifically to the nucleoid. How it's structured, how it's replicated, how it gets to opposite sides of the cells, before the cell divides, and how that is different from eukaryotes. So the nucleoid, is usually is typically condensed. It's where you have the circular chromosomal DNA. And also there's some proteins and RNA, histones and things like that. And it extends through the cytoplasm. All right, so bacterial DNA is organized in the nucleoid. Remember I told you today's Friday. Yeah, so Wednesday, I told you that we used to think that bacteria were just a bag of soup and everything was free floating, but the actual DNA is really nicely organized. And although it's not confined by a membrane, it's still pretty organized into its own domains. So the nucleoid forms chromosome loops called domains, which kind of radiate from the center. This right here is an electron micrograph. And so you can see right in B that they kind of just explode out. The chromosomes kind of explodes out when you lose that confinement. So it's not really a random pot of noodles. It does have to be confined. So that's something that, you know, we should make sure that we spread the word more as we move forward in the field. Questions? Great, you guys are all gonna ace that exam, aren't you? No. All right. One thing about bacteria is that when you go from DNA to RNA to protein, that's the central dogma of life. So DNA to RNA is transcription, RNA make proteins is translation. Those two processes are actually coupled, and that's something that's different from eukaryotes. So you have, you know, we're not going to go into the details of transcription and translation right now, but are you all familiar with transcription and translation and some of the enzymes involved? Okay, I just taught it in Bio 131 last semester, so I assume somebody had taught you that at some point. If you need a refresher, the book is great. But the RNA polymerase will transcribe the DNA into mRNA, that's transcription. And then that mRNA can immediately be bound by a ribosome for protein synthesis. So ribosomes are where proteins are made. And so that key there is that can kind of happen immediately after. And then, you know, there's another tRNA that brings amino acids to the ribosome that completes the translation. But what you should know is that in prokaryotes, transcription and translation are tightly coupled. Okay, so this is just kind of showing that things can, that they're coupled. You have your RNA polymerase over here. This is your DNA strand, and your RNA polymerase is moving along the DNA to make this mRNA in blue, and then the ribosome can hop onto it and start making the proteins or the peptide chains at the same time. And so this coordination of these processes explains why some bacteria can divide so quickly. It's because while you're making the mRNA, you could start making proteins, you know, along the same transcript. So that's different from eukaryotes. Bacteria coordinate these two processes. Eukaryotes separate them both spatially and temporally. So what that means is spatially means in space or in the certain organelles. Eukaryotes do the different processes in different organelles, and they don't happen at the same time. So they're separated temporally as well. Does that make sense to people? Okay. So cell division for bacteria is called binary fission. And so this requires, you know, really highly coordinated growth and expansion of all of the bacterial cell's parts. Unlike eukaryotes, prokaryotes synthesize RNA and proteins continually while the cell's DNA is undergoing replication. So before transcription, the DNA has to be replicated, and that's happening also simultaneously. And so bacterial DNA replication is coordinated with cell wall expansion and ultimately the separation of two daughter cells. So this is a nice picture kind of just explaining that. You have this origin of replication, or the OREC site, which is where the chromosome begins to replicate. And then you have, you know, these two replication forks that end up, you know, going in opposite directions. And so that is, you know, at each fork, you have basically a bunch of enzymes or proteins that are involved in that process, and we call that a replosome. So it's it's the complex of the replication fork, the DNA polymerase, other proteins that make that replication or replosome. And so we won't dive too much into this, but before dividing into two cells, you get the cell wall expansion. You know, we get this FTSZ protein that eventually allows it to split. And so for this ability for it to split, we call this septation. This is what completes the cell division. And usually you also have, just like an origin of replication, you have a termination site that triggers the growth of that dividing partition. So that's the septum. This is called the septum. So you guys should know that. It grows inwards and at last constricting and sealing off the two daughter cells. So if I were to ask what is the process that completes cell division, it would be septation. All right. So some specialized structures of bacteria, some bacteria, are thylakoids and carboxysomes. Thylakoids basically are part of bacteria that have carboxysomes. So carboxysomes are these organelles within certain bacteria that basically can photosynthesize. And so if you remember anything about photosynthesis and the thylakoid membrane being involved, this is also found among some bacteria. And so cyanobacteria are the typical example of bacteria that can photosynthesize. And so the thylakoids are just these extensively folded intracellular structures, and so they're also found in plant chloroplasts for photosynthesis. So some bacteria have thylakoids. Carboxysomes are these polyhedral bodies. They literally look like a polyhedral shape. And they are packed with the enzyme Rubisco. These are found in cyanobacteria, and they help fix carbon dioxide and to be utilized by other cells, and it converts carbon dioxide into sugar for the cells to grow. So this is an evolutionary mechanism that bacteria have evolved. The thylakoids and these carboxysomes. These are really cool. In my first year of grad school, I actually was rotating in a lab that studied the carboxysomes, and they did some really cool microscopy work with that. And Rubisco is an enzyme that's really important. It's actually, I don't know if you guys know this, but it is the most abundant enzyme on Earth. So cyanobacteria have contributed to a large amount of the oxygen production on Earth, as well as being able to fix sugars from carbon dioxide. And so having an abundant amount of Rubisco really helps with this process. Basically, the enzyme Rubisco sequesters the carbon dioxide, or it holds the carbon dioxide, and it's able to use it to make a lot of these sugars and oxygen. Questions? Okay. Sometimes it's actually good for some bacteria to adhere to surfaces, and so they do this by having pili or fimbriae. These are straight filaments of the protein pillin, and they're used in attachment. So they basically allow the cell to attach and adhere to surfaces so it doesn't move. These are, like string-like structures. These are different from the flagella, which is kind of the tail on some bacteria that allows for movement and motility. So these are used in attachment. A lot of people confuse pili for flagella. Another form of surface attachment is the structure stalks. Stalks are membrane-embedded extensions of the cytoplasm. So that helps them to attach to surfaces. And so the way that they do this, so this is the stalk, this is the bacteria, and this is the stalk. And they make this thing, it's like a glue, it's called holdfast. It allows it to adhere to the surface. So that secretion, it's an adhesion factor called holdfast that allow it to attach to surfaces. nanotubes are another type of intercellular connection. The key word here being intercellular, so they allow more than one cell to adhere to each other. So it's not to surfaces, it's to each other, and so that allows for different, material to be passed from one cell to the next. So you guys should know, the importance of these structures and just understand what they're made of, what they're used in, and if it's used in attachment, you should know what it's going to attach to. Okay, and then there are flagella. So usually if the environment is nutrient filled, it's good to stay put for bacteria. They don't need to move, but bacteria also live in these rapidly changing environments. So therefore, cell survival often necessitates the ability to move and relocate and reposition. So bacteria, not all bacteria, but a lot of bacteria are motile through these flagella. And so they basically have these helical propellers that drive the cell forward. So, you can see that these, similar to the Peelle in structure, but they're different in function. And so there are different types of bacteria with Peelle. You don't need to know all of these terms, but clearly monotrichous means you have one. Amphitrichous, you have two, they're on opposite sides. Lophotrichous, I guess, means you have a bunch that are on the back end, and then there's peritrichous, you have them kind of all over the cell. So yeah, they're randomly distributed around the cell. Flagella usually will rotate together in a bundle behind the swimming cell. So it's a rotary motion. There's literally like a motor in the back that rotates and allows for the bacteria to propel. Okay, and then this one's a cool one. It's a cool specialized structure called the magnetosome. And so basically it's an organelle inside of the membrane of bacteria that allows bacteria to navigate the Earth's magnetic field. So this is one of those newer-ish discoveries. They're basically able to align and navigate along the geomagnetic field of the Earth. And one of the ways that they do this is they're an aquatic bacteria, and they don't like oxygen. If they live, in water and they don't like oxygen, they find low oxygen environments by using this strategy. And so how do they do this? They form basically a compass needle inside of the cell. And so they do this in the form of magnetosomes, this organelle. So I think this is really cool. And yeah, they really help allow for, like, these are the magnetosomes right here, and so the way that these line up allows the cell to navigate and align to where it needs to go. Questions? The bacteria that have magnetosomes are called magnetotactic bacteria. They're found in a lot of aquatic environments. So they want to find low oxygen. Magnetite is biomineralized membrane-bound organelles. Okay, so I want to emphasize this because it's wrong in previous textbook. Whether you have or don't have organelles is no longer a difference between prokaryotes and eukaryotes. We now know that some bacteria have, diversity of different organelles, and they come in a variety of different flavors. The four that, you would need to know are these four different types of membrane-bound organelles that bacteria have. There are magnetosomes that are membrane-bound. We didn't talk about chromatophores and anamoxysomes, so I'm not gonna ask you anything on those, but you should know magnetosomes falls under membrane-bound organelles. Protein-bound organelles carboxysomes, that helical shape that I showed you guys, that's all a protein shell. You could have some that have lipid monolayers, and then there's phase defined. So we didn't talk much about these, so I wouldn't worry too much about these two, but all of these organelles, the purpose is to increase metabolic efficiency in certain chemical reactions, or to sequester specific compounds that are often toxic to the cell. So this is just, more than you need to know, but here we have the carboxysome, and so this whole shape is all protein. You see the Rubisco enzyme, and CO2 is coming into it, and so it's sequestering it. Okay, quick concept check, see if you guys are paying attention. What is an extension of the cytoplasm that attaches bacteria to a surface? Stalk. Yep. Remember, the main difference is that a stalk is a membrane enclosed extension of the cytoplasm, whereas, pili or fimbria help allow the cell to attach to a surface. All right. Any questions? Okay, well, we finished a little early today. Happy Friday.